Hello students, welcome to Vedantu Karnataka. Hegidira ellaru chennagidira. So we are going to do practice questions for FA1. So almost on 2 weeks inda ide maartta idivi. Some of them told that it was very helpful for them. Some of you might have first unit test later in the second half of August or maybe in the last week of August. So our time alli video nodak maribedi because this video has all the most important questions. Now where are we taking the questions from? We are taking the questions from the question banks that are given from the Karnataka PUC board. Okay, so board kadeinda kottiruva one set of questions now solve ಮಾಡ್ತಾ ಇದೀವಿ. So ಇವತ್ತೊಂದು small session let's do. I think it will be a very small session where I'll be solving the sexual reproduction in flowering plants topic. Some of the important questions that you have to study and go. Without this, you will not be able to score marks. Okay. So we are going to answer the following questions in 200 to 250 words each, wherever applicable. So 5-mark question no marana, 3-mark question. I will generally be taking main 5-mark questions because 5-mark questions are easy scoring. Alva, nimage easy score madbodu 5 marks question ge. So that's why you can always go for 5-mark questions. So I hope all of you smash the like button. Bega video go like kodi. Bega quickly smash the like button and then we can directly get into the video. Okay, ready? Okay, first question. Explain the structure of pollen grains. Okay, explain the structure of pollen grains. So pollen grains one by 10 students. First of all, nimagatta like you should be able to remember your color ella hoita. Okay, pollen grains are actually the male gametophyte. Okay, male gametophyte. Gametophyte andre eno, the ones that will have the pollen grains as the male gamete inside them, alva. So if they have the male gamete inside them, that means they're going to be called the male gametophyte. Now what have they asked? They've asked you the structure of a pollen grain. So how do you draw that? First ನೀವು ಒಂದು circle draw ಮಾಡಬೇಕು, which is going to be the outermost layer. Okay, so I'll draw it bigger so ನಿಮಗೆ clean ಆಗಿ explain ಮಾಡಬಹುದು. So here and then you draw outside. Okay, okay, we draw the outer layer which can be like this. Okay, because our outer layer banbit it's little ornamental in shape, that is because everything will have a little different shape. Okay, little different shape. So illi outer layer draw madidini. Okay, we're just shading it so it looks nice. Ee notes ella nimige Vedantu Karnataka Telegram channel alli irutte, illandre description box allu irutte. So don't worry about the don't worry about the notes. Now try and focus and try to learn here. So illi nodi idu vacuolization admele spindle fibers formation admele we are going to get 2 cells. Okay, we are going to get 2 cells. The first cell, which is the bigger cell. Okay, bigger cell one with students, vegetative cell. Okay, the bigger cell is called the vegetative cell, which will have a irregularly shaped nucleus. Okay, large shaped nucleus with dense cytoplasm. Okay, dots you have to draw. They'll have dense cytoplasm. Clear? Dense cytoplasm will be there, and down you will have a spindle shaped cell. Okay, with kind of like a smaller nucleus. Okay, smaller nucleus. Again, they will also have cytoplasm, but these people are going to have dense cytoplasm. Okay, in labeling nodana correct age. So this layer which I have drawn, this outer layer which I have drawn is called the exine. What is it called? The exine. So exine students, E for external, E for exine anta you can remember. So this outer layer which I have drawn is the exine. So I'm just bordering it because illandre nimage correct difference artha agala. Okay, 1 minute. You don't have to draw so beautifully in the exam, but whatever you draw, you have to do the labeling properly. So label manana, this outer layer is called as exine. So students, structure of pollen grains jote ನೀವು ಅದ ಬಗ್ಗೆ ಸ್ವಲ್ಪ if you write also there's no problem. So exine is made up of sporopollen. What is it made up of? Sporopollen, which is one of the most resistant substance. Okay, most resistant substance is going to be the sporopollen, and this is going to be called the intine. Okay, this is going to be called the intine, which is made up of cellulose and pectin. What is it made up of? Cellulose and pectin. ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಕರೆಕ್ಟ್ ಆಗಿ ಅರ್ಥ ಆಗುತ್ತೆ. ಎಕ್ಸೈನ್ ಬಂದ್ಬಿಟ್ಟು ಕಂಟಿನ್ಯೂಸ್ ಲೇಯರ್ ಅಲ್ಲ, ಅದೊಂದು ಡಿಸ್ಕಂಟಿನ್ಯೂಸ್ ಲೇಯರ್. ಡಿಸ್ಕಂಟಿನ್ಯೂಸ್ ಲೇಯರ್ ಲೀಡ್ಸ್ ಟು ಎನ್ ಅಪರ್ಚರ್. ಈ ಅಪರ್ಚರ್ ಬಗ್ಗೆನೂ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ ಮುಂಚೆ. ದಿಸ್ ಅಪರ್ಚರ್ ಇಸ್ ಆಕ್ಚುಲಿ ಕಾಲ್ಡ್ ದ germ pore, so that pollen tube germination can happen. Okay, pollen tube growth ಆಗಬೇಕಂದ್ರೆ because sporopollen ಬಂದು ತುಂಬಾ hard resistant substance, there is no way that the sporopollen is going to allow the pollen tube germination. So for pollen tube growth there is a small aperture which is called the germ pore. Okay, now going to the cell. Student, the larger cell is called the vegetative cell. Okay, the larger cell is called the vegetative cell, and remember that this vegetative cell is the one that is going to give all the reserve food material for the generative cell. Why? Because generative cell already male gamete presence is there. Okay, I hope all of you are understanding and marking your attendance. Okay, so vegetative cell has dense cytoplasm. Okay, has dense cytoplasm, and they have the reserve food material that will give all of it to the generative cell. Why, students? Because generative cell has the male gamete. Okay, generative cell has the male gamete. So generative cell. Artha, so this is the structure of a pollen grain. This is the structure of a pollen grain. This is very, very important for you. You can write here nucleus if you want. Nimagi ಇನ್ನೂ ಜಾಸ್ತಿ ಲೇಬಲ್ ಮಾಡಬೇಕಂದ್ರೆ ಅದು ಮಾಡ್ಬೋದು. ಓಕೆ, ನ್ಯೂಕ್ಲಿಯಸ್ ಅಂತ ಬರಿಬೋದು, ಸೈಟೋಪ್ಲಾಸಮ್ ಅಂತ ಬರಿಬೋದು. ಸೊ ದಿಸ್ ಈಸ್ ಅ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಡಯಾಗ್ರಾಮ್. ಕ್ಲಿಯರ್ ಟು ಆಲ್ ಆಫ್ ಯು? ಐ ಹೋಪ್ ಡಯಾಗ್ರಾಮ್ ಇಸ್ ಪರ್ಫೆಕ್ಟ್ಲಿ ಕ್ಲಿಯರ್. Done. Okay, awesome. Okay, next question nodana. Okay, this was the space given for writing it, never mind. Schematically represent the development of the female gametophyte. So ill nodi question noda time alli students, please try and understand the question. Okay, because schematic representation they have given. Okay, schematic representation, the development, okay, development of the female gametophyte. Female gametophyte first ಏನಂತ ಬರಿಬೇಕಿಲ್ಲಿ. Female gametophyte means they are talking about the embryo sac. Okay, they are talking about the embryo sac. So you have to draw the entire thing how the embryo sac formation is going to happen. So here please try and understand that whenever I am talking about the embryo sac, I have to start from what is. So embryo sac formation ಹೆಂಗಾಗೋದು? Because of megasporogenesis ಅಲ್ವಾ? Megaspore mother cell ಇಂದ megaspore tetrad form ಆಗುತ್ತೆ. Tetrad ಇಂದ one cell will become functional. That is going to undergo mitotic division and finally form the embryo sac. Yes or no? So if that formation is going to happen, I need to correctly draw the megasporogenesis process here. So I am going to start here, students, from the megaspore mother cell. Okay, megaspore mother cell, that is MMC. Okay, diploid agide. Okay, 2N, it is diploid. Now this megaspore mother cell or your MMC is going to undergo, okay, it is going to undergo the first meiosis. Okay, first meiosis. What is it going to form, students? This is going to form your megaspore tetrad. Okay, megaspore tetrad. ಈ megaspore tetrad ಅಲ್ಲಿ labeling ಮಾಡ್ತೀನಿ, megaspore tetrad. ಬಟ್ ಇಲ್ಲೊಂದು ಟ್ವಿಸ್ಟ್ ಇದೆ, ಸ್ಟೋರಿಗೊಂದು ಟ್ವಿಸ್ಟ್ ಇದೆ. ಇದು ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಥರ ಅಲ್ಲ. ಮೆಗಾಸ್ಪೋರ್ ಟೆಟ್ರಾಡ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, 3 ಜನ ಇಲ್ವಾ, ಅವರು ಡಿಸ್ಫಂಕ್ಷನಲ್ ಆಗುತ್ತೆ. ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬಿಕಮ್ ಡಿಸ್ಫಂಕ್ಷನಲ್, ಆ್ಯಂಡ್ ದ ಒನ್ ದಟ್ ಈಸ್ ಇನ್ ದ ಚಲಾಸಲ್ ಎಂಡ್, ಓಕೆ, ದ ಒನ್ ದಟ್ ಈಸ್ ಇನ್ ದ ಚಲಾಸಲ್ ಎಂಡ್ will undergo the development, and that kind of a development is called as monosporic development. Students, ನಾನು ಆಲ್ರೆಡಿ ಒನ್ ಶಾರ್ಟ್ ರಿವಿಷನ್ ಮಾಡಿದ್ದೀನಿ ಈ ಚಾಪ್ಟರ್ಗೆ. ಸೊ ನಿಮಗಿದೇನು ಅರ್ಥ ಆಗ್ತಾ ಇಲ್ಲ ಅಂದರೆ ಪ್ಲೀಸ್ ಆ ಒನ್ ಶಾರ್ಟ್ ರಿವಿಷನ್ ಹೋಗಿ ನೋಡಿ, ಬಿಕಾಸ್ ಅದರಲ್ಲೆಲ್ಲ ಕಾನ್ಸೆಪ್ಟ್ಸು ಕ್ಲಿಯರಾಗಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ. ಓಕೆ, so the one that is in the chalasal end, the megaspore, is going to continue the development, because only one spore is doing the development. Our development ನಾವು ಏನು ಕರಿತೀವಿ, monosporic development ಅಂತ ಕರಿತೀವಿ. Okay, monosporic development. Now students, after this monosporic development, what is going to happen? This one cell ilva, this one cell will undergo mitosis and it is going to do karyokinesis. Okay, so ಹಂಗೆ 2 cell ಆಗುತ್ತೆ, like 2 nucleus ಆಗುತ್ತೆ. One from 1 to 2, finally 2 to 4, 1, 2, okay, 3, 4, 4 to 8, 4 to 8. So this is the entire development that is going to happen. So let's say, we'll continue madana. So allindanu baribodu. So illi you write the megaspore. Okay, from the megaspore, one of them is going to undergo mitotic division, and it is going to become 2. Okay, students, please remember, ಇಲ್ಲಿ ಮೇನ್ಲಿ ಏನು ನಡೀತಾ ಇದೆ, ಕ್ಯಾರಿಯೋಕೈನೆಸಿಸ್ ನಡೀತಾ ಇದ್ರೆ ಅಂದರೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವಿಷನ್. ಓಕೆ, ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವಿಷನ್ ಇಸ್ ಹ್ಯಾಪನಿಂಗ್ ವಿತೌಟ್ ಸೈಟೋಕೈನೆಸಿಸ್, ಅಂದರೆ ದೇರ್ ಇಸ್ ನೋ ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್. ಅದಕ್ಕೆ ನಾವು ಈ ಥರ ನ್ಯೂಕ್ಲಿಯಸ್ ಕಂಟಿನ್ಯೂಸ್ಲಿ ಇಟ್ಸ್ ಫಾರ್ಮಿಂಗ್. ಸೊ ದಿಸ್ ವಿಲ್ ಲೀಡ್ ಟು ಹೌ ಮಚ್? This will lead to 4. 1, 2, 3, 4. Okay, 1, 2, 3, 4. So this is going to be the tetrad. Okay, or the not tetrad, you can write the 4 nucleate stage. 4 nucleate. Okay, don't be like, my god ma'am, from where are you writing all this? Is this like NEET level? Illa students, nim textbook allu ide 2 nucleate. Okay, 4 nucleate. Now from 4 nucleate, what is going to happen? Obviously 8 nucleate stage form agute. Okay, so 1, 2, 3, 4. Okay, 1, 2, 3, 4. ಇಲ್ಲಿಂದ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಫಾರ್ಮೇಷನ್ ಆಗೋದು. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ನಾವು ಆಲ್ರೆಡಿ ಬರಿತೀವಿ 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಅಂತ. ಓಕೆ, 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಅಂತ ಆಲ್ರೆಡಿ ಬರಿತೀವಿ. ನಾವು ಫ್ರಮ್ ಹಿಯರ್ ವಾಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಹ್ಯಾಪನ್? The formation is going to continue, that will lead to the embryo sac formation. ನಿಮಗೆ ಡೈರೆಕ್ಟ್ಲಿ embryo sac ಡಯಾಗ್ರಾಮ್ ಬರಿಬೋದು, ಇಲ್ಲಾಂದರೆ ಇದಾದಮೇಲೆ embryo sac ಅಂತಾನು ಬರಿಬೋದು, ಬಿಕಾಸ್ ನೀವು ಫುಲ್ ಎಲ್ಲ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀರಾ. ಬಟ್ ಸ್ಟೂಡೆಂಟ್ಸ್, embryo sac diagram is very very important. I'm just now going to draw like this. Okay, I'll explain you again separately. 2 cells in the center, ಇಲ್ಲಿ 2 cells ಇದೆ, ಆಮೇಲೆ ಇಲ್ಲೊಂದು ಸೆಲ್ ಇದೆ. ಓಕೆ, ದಿಸ್ ಇಸ್ ಯುವರ್ embryo sac formation. ಸೊ embryo sac ಬಂದ್ಬಿಟ್ಟು 7 cell 8 nucleated, 7 cell 8 nucleated ಇರುತ್ತೆ. ಓಕೆನಾ? ಸೊ 8 nucleated ಹಂಗೆ ಅಲ್ಲಿ ನೋಡಿ ಆಲ್ರೆಡಿ 8 nucleus ಇದೆ. 7 cell how? Because these cells ಬಂದ್ಬಿಟ್ಟು polar nuclei ಅಲ್ವಾ, we consider them as a single cell. Since we consider them as a single cell, it is going to be called as a 7 cell 8 nucleated state. Embryo sac, I'll explain you in detail again. Okay, I'll explain you in detail again. So this is clear, students? So schematic representation of the development of the female gametophyte. You start from the megaspore mother cell, will undergo meiosis, it will form the megaspore tetrad. Adalli 3 jana dysfunctional agutte, only one will continue the development. Our development ಗೆ ನಾವು monosporic development ಅಂತ ಕರಿತೀವಿ. ಅಲ್ಲಿಂದ stages continue ಆಗುತ್ತೆ. ಹೆಂಗಾಗುತ್ತೆ? 1 megaspore ಇಂದ 2 nucleate, 4 nucleate, 8 nucleate stage ಇಂದ ultimately ನಮ್ಮ embryo sac formation ಆಗುತ್ತೆ. Okay, finally embryo sac formation is going to happen. Clear? Is this clear to all of you? Yes or no? Bega thumbs up haki chat box alli. Come on. Done? Okay, good. Now next question nodana. Answer the following. Explain different types of pollination. Okay, different types of pollination based on the source of pollen. So pollination andre eno first ad ಗೊತ್ತಿರಬೇಕು. Okay, pollination andre, pollination means transfer of pollen grains. Okay, transfer of pollen grains from where, students? Where is the pollen grains going to be there? Anther. Very good. From anther to stigma, anther to stigma, transfer of pollen grains from anther to stigma of same plant irbodu, different plant irbodu. Okay, now ಇಲ್ಲಿ ನೋಡಿ same plant idre, okay, same plant means you call that as autogamy. ಏನದು? autogamy. Same plant idre. Okay, ಇವಾಗ same plant but different flower idre, ಕಾಣಿಸ್ತಾ ಇದೆಯಾ? different flower. So students, one plant alli one flower ಬಂದ್ಬಿಟ್ಟು female, ಇನ್ನೊಂದು flower male ಇದ್ರೆ same plant ಅಲ್ಲಿ 2 different flowers. Okay, so same plant different flowers is going to be called what? Geitonogamy. ಗಿಯಟ್ನೋಗ್ಯಾಮಿ. ಗಾಟ್ ಇಟ್? ಅದಾದಮೇಲೆ ಇಲ್ಲಿ ಸೇಮ್ ಪ್ಲಾಂಟ್ ಅಲ್ಲ, ಕಂಪ್ಲೀಟ್ಲಿ ಡಿಫ್ರೆಂಟ್ ಪ್ಲಾಂಟ್ ಇದ್ರೆ, ಓಕೆ, ಡಿಫ್ರೆಂಟ್ ಪ್ಲಾಂಟ್, ವಿ ಕಾಲ್ ಇಟ್ ವಾಟ್? We call it xenogamy. So xenogamy andre we can also call in simple words cross pollination. Okay, xenogamy andre namak simple agi cross pollination antanu karibodu. Second question ಏನು ನೋಡಿ, list the characteristic feature of water pollinated flowers. So water pollinated flowers characteristic features ಏನಂತ. So ಇಲ್ಲಿ diagram draw ಮಾಡಿದೀನಿ. Autogamy ಬಂದ್ಬಿಟ್ಟು self pollination, transfer of pollen grains from anther to stigma of the same flower. ಗಿಯಟ್ನೋಗ್ಯಾಮಿ ಅಂದರೆ ಒಂದೇ ಪ್ಲಾಂಟಲ್ಲಿ 2 ಡಿಫ್ರೆಂಟ್ ಫ್ಲವರ್ಸ್ ಇದೆ ನೋಡಿ, ಮೇಲು ಇದೆ ಫಿಮೇಲು ಇದೆ. ಸೊ ಈ ಫ್ಲವರಿಂದ ಈ ಫ್ಲವರ್ಗೆ ಹೋಗ್ತಾ ಇದೆ, ಬಟ್ ಸೇಮ್ ಪ್ಲಾಂಟ್ ಸ್ಟೂಡೆಂಟ್ಸ್. ಒಂಥರ ನಿಮ್ಮ ಇದು ಮದುವೆ ಮಾಡ್ತಾರಲ್ವಾ ಇನ್ಸೈಡ್ ಫ್ಯಾಮಿಲಿಯಲ್ಲಿ, ಲೈಕ್ ಮಾಮ ಪೊಣ್ಣು, ಅತ್ತೆ ಪೊಣ್ಣು, ಮಾಮ ಪಯ್ಯ, ಸೊ ಆ ಥರನೇ ಸೇಮ್ ಫ್ಯಾಮಿಲಿ, ಬಟ್ ಆದರೆ ಕಸಿನ್ಸು, ಫಸ್ಟ್ ಕಸಿನ್ ಸೆಕೆಂಡ್ ಕಸಿನ್ ಮದುವೆ ಮಾಡೋದು. ಬಟ್ ಆದರೆ ಕೂಡ ಫ್ಯಾಮಿಲಿಯಲ್ಲಿರೋ ಎಲ್ಲ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸು, ಎಲ್ಲ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸು ಸಿಮಿಲರ್ ಆಗೇ ಇರುತ್ತೆ, ಅಷ್ಟು ವೇರಿಯೇಷನ್ ಇರಲ್ಲ ಅಲ್ಲ. ಸೊ ಇಲ್ಲಿ ಆಲ್ಮೋಸ್ಟ್ ನೋ ವೇರಿಯೇಷನ್. ಇಲ್ಲಿ ನೋಡಿ xenogamy ಅಂದರೆ ಫ್ರಮ್ ಒನ್ ಫ್ಲವರ್ ಟು ಅನದರ್ ಫ್ಲವರ್, anther to stigma of different flower, complete variation will be there. It's like you are marrying from someone separate. Okay, you don't even know those family, but you are getting married to them. So their characteristics is going to be completely different. Clear to all of you? Yes or no? Okay, awesome. So now they have asked, what is the characteristic feature of the water pollinated flowers? So in the NCERT, so illi idu first portion alla beda, illi nodi some examples of water pollinated plants are Vallisneria and Hydrilla. Okay, Hydrilla, which grow in fresh water, and several marine sea grasses called as Zostera. Okay. Now, not all aquatic plants use water for pollination in majority. So, water hyacinth matte water lily, water use madalla pollination ge. They wait for what, students? They wait for insect illandre wind for pollination. Adontara eno ondu assurity, because avrige artha aitu that water pollination banbit ashten doddu feature alla. Nanige already nan ಏನು ಮಾಡ್ತೀನಿ, I'll just come above the water level and I will just wait for insect or wind to do pollination. So that is why water hyacinth and water lily is doing that. Clear? Now, in Vallisneria, the female flowers reach the surface of water by long stalk. So when they are asking you characteristics, okay, when they are asking you characteristics, students please ನಿಮ್ಮ exam dates comment ಅಲ್ಲಿ ಸ್ವಲ್ಪ ತಿಳಿಸಿ, like when is your exams, when is your first unit test anta. So according to that we can help you prepare. So look here, they're saying that in Vallisneria they are going to have long stalk. So ad baribodu characteristic, they're going to have long stalk. Okay, long stalk, and the male flowers of the pollen grains are released on the surface of water. They are carried passively by water currents. Some of them eventually reach the female flower and the stigma. In other group of water pollinated plants such as sea grasses, female flowers remain submerged in the water and the pollen grains are released inside the water. Pollen grains in many such species. So nodi pollen grains characteristic kelidare process alla. So id ಬಗ್ಗೆ ಬರಿಬೇಕು. Pollen grains are what? Long ribbon like, nodi long and ribbon like. Okay, long and ribbon like they will be, and passively some of them reach the stigma. In most water pollinated species, pollen grains are protected. So pollen grains are going to be protected by a mucilaginous coating. Pollen grains protected by mucilaginous covering. Clear? Mucilaginous covering. These are the characteristics of your water pollinated flowers. Idella baribodu like Vallisneria, Zostera, adella example baribodu. Like example, don't write water hyacinth and water lily because avaru water pollination madalla. So here you can write Vallisneria, okay, Vallisneria, and you can write Hydrilla, and Zostera, that is seagrass. You can write these as the example, okay. ಈ ನೋಟ್ಸ್ ಎಲ್ಲ ನಿಮಗೆ ಸಿಗುತ್ತೆ. ಈ ನೋಟ್ಸ್ ಎಲ್ಲ ನಿಮಗೆ ವೇದಾಂತು ಕರ್ನಾಟಕ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲಲ್ಲೂ ಮತ್ತು ಡಿಸ್ಕ್ರಿಪ್ಷನ್ ಬಾಕ್ಸಲ್ಲೂ ಇರುತ್ತೆ. ಓಕೆನಾ? ಸೊ ಮೋಸ್ಟ್ಲಿ ಡೈರೆಕ್ಟ್ಲಿ ಟೆಲಿಗ್ರಾಮ್ಲೇ ಶೇರ್ ಮಾಡ್ತೀನಿ ಬಿಕಾಸ್ ಅಗೇನ್ ನಾವು flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination commented. So, ಇದೊಂದು ಕ್ವೆಶನ್. ಸೊ ಬೇಸಿಕ್ಲಿ ಈ ಕ್ವೆಶನ್ನ ಫಸ್ಟ್ ಡಿಕೋಡ್ ಮಾಡಿ ಏನು ಹೇಳ್ತಾ ಇದ್ದಾರೆ, ಡಿಸ್ಕರೇಜ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ದಟ್ ಮೀನ್ಸ್ ವಾಟ್, ಅವರು ಇನ್ಬ್ರೀಡಿಂಗ್ ಡಿಪ್ರೆಷನ್ ಹೌ ಟು ಅವಾಯ್ಡ್ ಇಂಬ್ರೀಡಿಂಗ್ ಡಿಪ್ರೆಷನ್ ಅಂತ ಅವ್ರಿಗೆ ಗೊತ್ತು. ಕಂಟಿನ್ಯೂಸ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ವಿಲ್ ಲೀಡ್ ಟು ಇಂಬ್ರೀಡಿಂಗ್ ಡಿಪ್ರೆಷನ್. ಈ ಇಂಬ್ರೀಡಿಂಗ್ ಡಿಪ್ರೆಷನ್ನ ಡಿಸ್ಕರೇಜ್ ಮಾಡೋದಕ್ಕೆ ದೇ ಹ್ಯಾವ್ ಫೌಂಡ್ ಸಮ್ ಔಟ್ ಬ್ರೀಡಿಂಗ್ ಡಿವೈಸಸ್. What have they found? Some outbreeding devices. So they are asking, write about the outbreeding devices. So when you write about outbreeding devices, ಏನು ಬಗ್ಗೆ ಬರೀತೀರಾ students? You are going to write the first outbreeding device that a plant have taken care. ಅಂದರೆ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಅವಾಯ್ಡ್ ಮಾಡಬೇಕಂದ್ರೆ ಏನು ಮಾಡಬೇಕು, ನಮಗೆ ಫಸ್ಟ್ ಸಿಂಪಲ್ ಆಗಿರುವ ಯುನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್ ಫಾರ್ಮೇಷನ್ ಬರೀಬೋದು. Okay, you can write one word and you can maybe explain a little, no problem in that. Unisexual flowers, that means what, unisexual flowers formation is going to happen where they will form unisexual flower. On one plant there will be male flower, okay, on one plant there will be male flower and on another plant there will be female flower. Clear? This is the formation of unisexual flowers. Now 2, okay, second one the students, difference in length, okay. So difference in length of anther or stigma. That means what, normally ಹೆಂಗಿತ್ತು, normally situation ಹಿಂಗಿತ್ತು where ನಮಗೆ ಸ್ಟಿಗ್ಮಾ ಈ ಥರ ಲ್ಯಾಂಡಿಂಗ್ ಪೋರ್ಷನ್ ಅಲ್ವಾ, ದಿಸ್ ಇಸ್ ಹೌ ದ ಸ್ಟಿಗ್ಮಾ ಯೂಸ್ ಟು ಬಿ ದೇರ್. ಇಲ್ಲಿಂದ ಆಂಥರ್ ಬರ್ತಾ ಇತ್ತು ಆ್ಯಂಡ್ ಇಟ್ ಯೂಸ್ ಟು ಬಿ ಅಬೌವ್ ದ ಸ್ಟಿಗ್ಮಾ. ಸೊ ಈ ಥರ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಫಾಲ್ ಮಾಡೋದು ತುಂಬ ಈಸಿ ಆಗಿತ್ತು. ಸೊ ಅದಕ್ಕೆ ಇಲ್ಲಿ ಏನು ಮಾಡಿದ್ದಾರೆ, ಫ್ಲವರ್ಸು ಲೆಟ್ಸ್ ಸೇ ಸ್ಟಿಗ್ಮಾ ಅದೇ ಹೈಟಲ್ಲಿದೆ, ಓಕೆ, ಮೇ ಬಿ ಅವರು ಫಿಲಮೆಂಟ್ ಹೈಟ್ ಸ್ವಲ್ಪ ಕಮ್ಮಿ ಮಾಡ್ತಾರೆ. ಸೊ ಇದಕ್ಕಿಂತ ವಾಟ್ ಈಸ್ ಗೋನ್ ಹ್ಯಾಪನ್, ಬಿಕಾಸ್ ಆಫ್ ದಿಸ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಈಸಿಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗೋಕ್ಕೆ ಚಾನ್ಸಸ್ ತುಂಬ ಕಮ್ಮಿ ಇದೆ. ಓಕೆ, ಡಿಫ್ರೆನ್ಸ್ ಇನ್ ಲೆಂತ್ ಆಫ್ ಆಂಥರ್ ಆ್ಯಂಡ್ ಸ್ಟಿಗ್ಮಾ. ನೆಕ್ಸ್ಟ್ 2 ರೀಸನ್ಸ್ ಏನಂದರೆ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇಲ್ಲಿ ನೋಡಿ, ಥರ್ಡ್ ಒನ್ ಬಂದ್ಬಿಟ್ಟು ರಿಸೆಪ್ಟಿವಿಟಿ, ಚೇಂಜ್ ಇನ್ ರಿಸೆಪ್ಟಿವಿಟಿ. ಅಯ್ಯೋ, ರಿಸೆಪ್ಟಿವಿಟಿ, ಚೇಂಜ್ ಇನ್ ರಿಸೆಪ್ಟಿವಿಟಿ ಅಂದರೆ ಲೆಟ್ಸ್ ಸೇ ಫಸ್ಟ್ ಏನಾಗುತ್ತೆ, ಲೆಟ್ಸ್ ಸೇ ಫಸ್ಟ್ maybe first anther will mature, okay, first anther will mature, then first anther matures, then stigma or vice versa. ಅಂದರೆ changing the receptivity, both of them, if they don't become active at the same time, okay, let's say a couple wants to conceive, okay, then both of them have to be in the reproducible age. ಆ ಥರನೇ ಸ್ಟಿಗ್ಮಾನು ಮತ್ತು ಆಂಥರ್, ಓಕೆ, ಅವ್ರು ಎರಡು ರಿಸೆಪ್ಟಿವಿಟಿ ಬಂದ್ಬಿಟ್ಟು ಸೇಮ್ ಟೈಮಲ್ಲಿ ಇರಬೇಕು. ಇಲ್ಲಾಂದರೆ ಆಂಥರ್ ವಿಲ್ ಪುಟ್ ದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಬಟ್ ಸ್ಟಿಗ್ಮಾ ವಿಲ್ ಬಿ ಲೈಕ್ ಐ ಡೋಂಟ್ ಕೇರ್. ಓಕೆ, ಆ ಥರ ಒಂದು ಡಿಫ್ರೆನ್ಸ್ ಮಾಡ್ತಾರೆ ಸೊ ದಟ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಡಸ್ ನಾಟ್ ಹ್ಯಾಪನ್. ಲಾಸ್ಟ್ ಬಂದ್ಬಿಟ್ಟು ಸ್ಟೂಡೆಂಟ್ಸ್, ಸೆಲ್ಫ್ ಇನ್ಕಂಪ್ಯಾಟಬಿಲಿಟಿ. Self incompatibility. That means what? That means rejecting the own pollen grains. Rejecting the own pollen grains. Now let's say you are telling I don't want to marry anyone inside my family. I want someone who is outside the family. So andre ಏನು, ನೀವು ನಿಮ್ಮ ಮನೆ ಒಳಗಡೆ ಎಲ್ಲಾನು ರಿಜೆಕ್ಟ್ ಮಾಡಿದ್ದೀರಾ. Okay, like let's say someone cousins or whatever. So ಆ ಥರನೇ self incompatibility. That means trying to be incompatible with the own pollen grains. So ಇಲ್ಲಿ NCERT is given, so let's go through that as well. So ಇಲ್ಲಿ ನೋಡಿ, continued self pollination results in inbreeding depression. Okay, to avoid inbreeding depression there is to encourage cross pollination. In some species what is going to happen students, in some species, first point ಇಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ, some species pollen, okay, pollen releases and stigma's receptivity are not synchronized. So, their receptivities are not synchronized. Next one, either the pollen is released before the stigma becomes receptive, or the stigma becomes receptive much before the pollen. In some other species, what is going to happen? The anther and the stigma are at different positions. Okay, so length wise, illandre position wise, our change agirutte. So, illu position word use madana, because NCERT what it is given, difference in position of anther or stigma. Okay, of anther or stigma. Now, both of these devices prevent autogamy, so that the pollen cannot come in contact with the stigma. Third device, students, prevent inbreeding is self-incompatibility. Okay, ನಾನು ಅಂಡರ್ಲೈನ್ ಮಾಡಿಲ್ವಾ ಆ ಥರನೇ ನಿಮ್ಮ NCERT ಅಲ್ಲೂ ಅಂಡರ್ಲೈನ್ ಮಾಡಿ. ಸೊ ವೆನ್ ಯು ಆರ್ ಡೂಯಿಂಗ್ ರಿವಿಷನ್ ನಿಮಗೆ ಈಸಿಯಾಗಿ ಓದ್ಬೋದು. This is a genetic mechanism and prevents self-pollen from fertilizing the ovules by inhibiting it. Another device to prevent is the formation or the production of unisexual flowers. Clear? Unisexual flowers. So if both male and female flowers are present on the same plant, such as in castor and maize, it prevents autogamy but gaietnogamy agutte. Because why? Same plant alli male female idre illinda il tanka release agutte, variation ashtu jaasti iralla. So that means they are telling that, let's say the plant, okay, let's say the plant has male and female flowers separate, but in the same plant, then it is not going to avoid anything. It is only going to avoid autogamy, it will not avoid gaietnogamy. Gaietnogamy avoid ಮಾಡಬೇಕಂದ್ರೆ male flower female flower separate plant ಅಲ್ಲಿ ಇರಬೇಕು. Okay, separate plant. So that's why they are telling in the last. So these are the 4 points. You please remember. Point number 1, 2, 3, okay, and 4. 4 outbreeding devices you have to remember and keep, so that this becomes easy, okay, to understand that what is this and how are they going to avoid it. Okay, how are they going to avoid it? Cleared so far? Okay, now double fertilization is unique event in flowering plants. Briefly explain. Arrange the following terms. First namige following terms na arrange madana, adamele we will go to the first question. Okay, so ಇಲ್ಲಿ ನೋಡಿ 3 marks and 2 marks. Okay, arrange the following terms in correct developmental sequence. Pollen grains is there, sporogenous tissue is there. So what is this? To arrange, they have given the microsporogenesis process. Microsporogenesis process. Okay, process they have given. Microsporogenous process they have given. So il nodi, we start obviously from the mother cell. Okay, so pollen mother cell. Okay, pollen mother cell, which is called as PMC. Okay, PMC, pollen mother cell. Pollen mother cells, pollen mother cell munche namak sporogenous tissue baribeko. So sporo, one second, can I, one second, can't spell it, I will note the correct aga. Okay, sporo, correct, no? Yeah, sporogenous tissue. Okay, sporogenous tissue will form the pollen mother cell. Pollen mother cells are going to undergo meiosis, students, and what are they going to form? They are going to form the microspore tetrad. Okay, they are going to form the microspore tetrad. Microspore. So ನಿಮಗೆ full microsporogenesis ಈ ಒಂದು flowchart ಇಂದ ಓದಬಹುದು. Microspore tetrads. Microspore tetrads will form what, students? Each of them is going to become a pollen grain. Okay, so illinda pollen grain formation. And pollen grains olagade ಏನು ಇರುತ್ತೆ? Generative cell will have the male gamete. Okay, generative cell will have the male gamete. Clear? Generative cell will have the male gamete. So flowchart is correct. How many they have given us? 1, 2, 3, 4, 5. Okay, 1, 2, 3, 4, 5. So sporogenous tissue, they are potential. They have the ability to become the pollen mother cell. So they will become the pollen mother cell and they will form the microspore tetrad. Microspore tetrad will form the pollen grains. Pollen grains will form the male gamete. Clear to all of you? Yes or no? Yes? Done? Okay, awesome. Now, first question. Okay, first question we have to understand. So students, first question. Okay. First question talks about double fertilization. Okay, what is double fertilization? Come on in the chat. Yellaru. Double fertilization. So attendance mark marta idira. Are we marking the attendance? Tell me quickly. Okay. So double fertilization means, okay, double fertilization means, students, here two types of fertilization is happening. Okay. So double fertilization alli enagutte, syngamy plus triple fusion nadiyutte. Syngamy plus triple fusion. Syngamy plus triple fusion nadiyutte. ಇವಾಗ syngamy plus triple fusion ಅಂದ್ರೆ ಏನು? syngamy ಅಂದ್ರೆ zygote formation. Okay, in syngamy zygote formation will happen. In triple fusion PEN, primary endosperm nucleus, ಇಲ್ಲಾಂದ್ರೆ simple words ಅಲ್ಲಿ endosperm ಅಂತ ಬರೀಬಹುದು. it formation ಆಗುತ್ತೆ. ಬಟ್ ಮ್ಯಾಮ್ ಇದು ಹೆಂಗಾಗೋದು ಮ್ಯಾಮ್? ವಾಟ್ ಇಸ್ ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂದರೆ ಫಸ್ಟ್ ನೀವು ಒನ್ ಶಾರ್ಟ್ ವಿಡಿಯೋ ನೋಡಬೇಕು. ಇಲ್ಲಿ ನೋಡಿ, ಬೇಸಿಕ್ಲಿ ಏನಾಗುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್, ನಾರ್ಮಲಿ ನಮ್ಮ ಪೋಲನ್ ಗ್ರೇನ್ಸಿಗೆ ಒನ್ ಮೇಲ್ ಗ್ಯಾಮಿಟ್ ಇರುತ್ತೆ. ದಟ್ ಮೇಲ್ ಗ್ಯಾಮಿಟ್ ವಿಲ್ ಗೋ ಆ್ಯಂಡ್ ಫ್ಯೂಸ್ ವಿತ್ ದಿ ಎಗ್ ಅಲ್ವಾ? ಬಟ್ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, ದ ಜನರೇಟಿವ್ ಸೆಲ್, ಓಕೆ, ಜನರೇಟಿವ್ ಸೆಲ್ ಇಲ್ವಾ, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಒಳಗಡೆ ಇದ್ದ ಜನರೇಟಿವ್ ಸೆಲ್ ವಿಲ್ ಅಂಡರ್ಗೋ ಮೈಟೋಸಿಸ್. ಓಕೆ, ವಿಲ್ ಅಂಡರ್ಗೋ mitotic division, and ಈ mitotic division ಇಂದ ಏನು ಫಾರ್ಮ್ ಆಗೋದು? ಎರಡು ಮೇಲ್ ಗ್ಯಾಮಿಟ್ಸ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ನಾರ್ಮಲಿ only one male gamete would be formed. Now here two male gametes are going to be formed. Now this two male gametes to go, like two boys is there means two brides should be there, no? So two male gametes are there. So what is going to happen here? 1 mg, okay, 1 mg andre one male gamete plus egg ilva, embryo sac olagade ira egg will fuse to form zygote. Okay, this process will lead to the formation of a diploid cell. Okay, it will lead to the formation of a diploid cell. This is normal scenario. Okay, this double fertilization, students, mainly agadalli in a 3-celled angiosperm. Okay, that means 2-celled angiosperms and 3-celled angiosperms will be there. This is 3-celled angiosperm. Why? Because generative cell will form 2 male gametes and normally one vegetative cell will be there. So it is like 3-celled, okay, 3-celled angiosperms. Now look here, 1 mg plus egg will give you the zygote, and inon mg ilva. So one more mg will fuse with what? Polar nuclei. Will fuse with the polar nuclei, and it will form the primary, it will form what? Primary endosperm nucleus. Okay, that will be endosperm. Okay, endosperm, primary endosperm nucleus that lead to the formation of endosperm. Clear to all of you? Easy agidya? Is it easy? So it will form 1 mg, that is plus polar nuclei, will form PEN, primary endosperm nucleus. Okay, primary endosperm nucleus. Now look here, that is your double fertilization. So either pollen grains will come. Okay, it will have the 2 cells. Can you see 2 cells? Because generative cell will undergo mitotic division. Here male gametes form agute. One male gamete will go and fuse with the egg. One more male gamete will go and fuse with the polar nuclei. So double fertilization andre syngamy matte triple fusion. One male gamete plus egg gives zygote. One male gamete plus polar nuclei gives endosperm. Illandre primary endosperm nucleus. Clear to all of you? Easy. So all formation agutte students. Illon triploid formation agutte. Okay, this is a triploid. Triploid formation agutte. So you can remember triploid and triple fusion. Triploid and triple fusion you can easily remember. Is this easy? Easy, right? Chalo, okay. Okay, a picture of a mature angiosperm embryo sac is given below. Answer the following question. First labeling marna, adamel answer marna. Okay, open your NCERT. Ivaga idu clear agilla andre NCERT open madi. So students, the cells which are towards the chalazal. Okay, idu chalazal. This is the chalazal end. This is the micropylar end. Okay, this is the micropylar end. This is the chalazal end. So E cells this is called as the antipodals. Okay, this is called as what? The antipodal. So AC anta remember madi, antipodal cells are towards the chalazal end. Okay, amele ide students ಎರಡ್ circle ilva. This is called as the polar nuclei. polar nuclei. Okay, then we will have the central cell. Okay, central cell. Clear? Central cell. Then this is going to be called the egg. Okay, egg. And this is the synergids. Okay, synergids. And synergids alli illi ondo finger like projections ide which are called as filiform apparatus. Why do we call them filiform apparatus and what is their function, students? ಅವರು ಪೋಲೆನ್ ಟ್ಯೂಬ್ಗೆ ಗೈಡ್ ಮಾಡ್ತಾರೆ, ಈ ರೋಡಲ್ಲಿ ಬನ್ನಿ ಅಂತ ಆ ಥರ ಗೈಡ್ ಮಾಡ್ತಾರೆ. ಸೊ ಫಿಲಿಫಾರ್ಮ್ ಅಪ್ಯಾರಿಟಸ್. ಈ ಸಿನರ್ಜಿಸ್ ಮತ್ತೆ ಎಗ್, ಓಕೆ, ದಿಸ್ ಟುಗೆದರ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದಿ ಎಗ್ ಅಪ್ಯಾರಿಟಸ್. ಇಟ್ಸ್ ಟುಗೆದರ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ಎಗ್ ಅಪ್ಯಾರಿಟಸ್. ಕ್ಲಿಯರ್? So this is the labeling of your embryo sac. Innone cell is a central cell. Okay, I have drawn everything. So this is the labeling of your embryo sac. Clear? Done. Okay, now next question. Which, okay, our same embryo sac question in the continuation. Which cells or nuclei of the embryo sac produce zygote and primary endosperm nucleus? So zygote formation. So which cells or nuclei of the embryo sac anta kelidare. So zygote formation bandu yaar form madodu? Egg, okay, egg matte male gamete. Okay, egg and male gamete, correct? So zygote formation will be taken care by egg and male gamete. Here what is going to happen? Polar nuclei plus male gamete. Whatever you wrote about double fertilization, you write it here. Clear? Now, what is the ploidy of antipodal cells in primary endosperm nucleus? Ploidy, here it is going to be, students, N anta irutte ploidy. Okay? Everything is going to be N. Ploidy is going to be N. Adamela avaru kelirodu primary endosperm nucleus ploidy ಏನಂತ. Primary endosperm nucleus ploidy ಬಂದ್ಬಿಟ್ಟು triploid ಆಗಿರುತ್ತೆ. 3N ಅಂತ ಬರುತ್ತೆ. Because you already know N plus 2N ಅಲ್ವಾ? One male gamete plus polar nuclei will give you 3N. Now, why the endosperm development precedes the embryo development? Precedes andre, why does endosperm gets formed first? Adamele embryo formation agadu. Why? Because, students, endosperm is the reserve. It has the it has the reserve food material. Okay, it has the reserve food material. So obviously it will give the nourishment for the growing embryo. Okay, it provides and the baribeko, it provides the nourishment for the growing embryo. Clear? So that is why this should be formed first, then the other one should be formed. Alva? Ilandre, how does it make sense? Right? How does it make sense? If one only forms, then the other also should form, right? So that is what the formation is all about. Clear? All of you, quickly smash the like button also. Bega. Okay, done. Okay, so nourishment for the growing embryo. Done. Now illon question nodi. Banana is parthenocarpic fruit, whereas orange is shows polyembryony. How are they different from each other with respect to seeds? Okay, banana is parthenocarpic fruit and orange shows polyembryony. So difference enu ille? They are asking in respect to seeds ಬಗ್ಗೆ ಬರಿಬೇಕು. Okay, banana is parthenocarpic fruit and it is developed without fertilization. That means this is fruit formed, okay, without fertilization. Okay, fruit formed without fertilization. Illi nodi, orange shows polyembryony, whereas multiple embryos are formed in a single seed. So it contains multiple embryos in one seed. So ill one seed ide, but ill nodi seedless. Okay, this is seedless, this has one seed. So orange ಏನಾಗುತ್ತೆ students, here we have multiple embryos. Okay, multiple embryos, and this embryo formation is because of the nucleus cells. Okay, already explained ಮಾಡಿದೀನಿ NCERT ಅಲ್ಲಿ ಇದೆ ಅಲ್ವಾ, that nucleus cells will come front and they'll be like, I want to be the embryo. ಆ ಥರ ಒಂದು setup ಇದು. Okay, multiple embryos. Done, Renadan? Okay, ಇವತ್ತಿಷ್ಟೇ ಕ್ವೆಶನ್ಸ್ ಇರೋದು. ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಅದು ಮೋಸ್ಟ್ಲಿ ಟುಮಾರೋ ದ ಕ್ಲಾಸ್ ವಿಲ್ ಬಿ, ಸೊ ದಟ್ ಟೈಮ್ ಐ ವಿಲ್ ಡೂ ಮೋರ್ ಕ್ವೆಶನ್ಸ್ ವೆರ್ ಐ ವಿಲ್ ಬಿ ಎಕ್ಸ್ಪ್ಲೇನಿಂಗ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್, ಓವ್ಯೂಲ್ ಡಯಾಗ್ರಾಮ್, ಆ ಥರ ಸ್ವಲ್ಪ ಟಾಪಿಕ್ಸ್ ಇದೆ. ಸೊ ಅದೆಲ್ಲ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. ಓಕೆ ಸ್ಟೂಡೆಂಟ್ಸ್, ಸೊ ಇಫ್ ಯು ನ್ಯೂ ಟು ದ ಚಾನಲ್, ಫಸ್ಟ್ ಟೈಮ್ ಇಫ್ ಯು ಸೀಯಿಂಗ್ ಮೀ, ದಟ್ ಮೀನ್ಸ್ ಯು ಹ್ಯಾವ್ ನಾಟ್ ಸಬ್ಸ್ಕ್ರೈಬ್ ಟು ದ ಚಾನಲ್. ಸೊ ಬೇಗ ಚಾನಲ್ಗೆ ಒಂದು ಸಬ್ಸ್ಕ್ರೈಬ್ ಹೊಡೀರಿ ಬೇಗ. ಓಕೆ, ಆ್ಯಂಡ್ ಲೆಟ್ಸ್ ಮೇಕ್ ದಿಸ್ ಚಾನಲ್ 40K. ಐ ಥಿಂಕ್ ಆಲ್ ಆಫ್ ಯು ಕ್ಯಾನ್ ಡೂ ಇಟ್. ಓಕೆ, ಸೊ ಬೇಗ ಲೆಟ್ಸ್ ಬಿಕಮ್ 40K. ಆ್ಯಂಡ್ ಆಲ್ಸೋ ನೋಟ್ಸ್ ಎಲ್ಲ ನಿಮಗೆ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಇರುತ್ತೆ, ಡಿಸ್ಕ್ರಿಪ್ಷನ್ ಬಾಕ್ಸಲ್ಲಿ ಲಿಂಕ್ ಇದೆ. ಜಸ್ಟ್ ಗೋ ಡೌನ್, ಜಾಯ್ನ್ ಆರ್ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಅಂತ ಇರುತ್ತೆ, ಕ್ಲಿಕ್ ಆನ್ ದಟ್ ಆ್ಯಂಡ್ ಯು ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಜಾಯ್ನ್ ಆರ್ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್. ಓಕೆ, ಸೊ ಥ್ಯಾಂಕ್ ಯು ಸೊ ಮಚ್. ಸಿ ಯು ಆಲ್ ಇನ್ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್. ಟಿಲ್ ದೆನ್ ಟೇಕ್ ಕೇರ್, ಬೈ.